Hello everyone and welcome back to Easy Dentistry. Our motto to start with this channel is to make lectures easy for students worldwide. All the lectures on our channel Easy Dentistry are absolutely free. There are no paid services on any lectures. Just you have to do one thing, that is subscribe to our channel and hit on the bell icon so that whenever we upload a new lecture, you get a notification of it. Please students, help us reach the 20,000 subscribers milestone soon. So subscribe right now. So today's lecture is a very important topic, that is aminoglycoside antibiotics. This is a very important question which is asked in exam. So watch this lecture carefully. So in this lecture, we will be studying the introduction to aminoglycosides. What is the classification of aminoglycosides, the mechanism of action, mechanism of resistance, the shared toxicities of aminoglycosides, then the precautions to be taken while administering aminoglycosides and the interaction of aminoglycosides with various drugs. Then we will be studying the pharmacokinetics. And then we would be studying each aminoglycoside in detail, namely gentamicin, streptomycin, kanamycin, tobramycin, amikacin, sizomycin, and neomycin, neomycin, and framycetin. So starting with the introduction, aminoglycosides are a group of natural and semi-synthetic antibiotics. They have a polybasic amino group which is linked glycosidically to two or more amino sugar residues. The aminoglycosides are used extensively in medical practice and they are seldom used in dentistry. These aminoglycosides are the products of deliberate research for drugs effective against the gram-negative bacteria. The first discovered aminoglycoside was streptomycin, which was discovered in 1944 by Waxman and his colleagues. All the aminoglycosides are derived from the actinomycetes, soil actinomycetes. So, as they are derived from a common source, the aminoglycosides share many common properties. Now, the classification of aminoglycosides. The aminoglycosides are derived into systemic ones and the topical ones. The systemic ones include streptomycin, gentamicin, kanamycin, tobramycin, amikacin, sizomycin, neomycin, while the topical aminoglycosides include the neomycin and the framycetin. Now let us study the mechanism of action of aminoglycosides. The action is mainly bactericidal and it can be described in two steps, step 1 and step 2. Now let us study in detail what are these steps. In the step 1, there is transport of the aminoglycoside through the bacterial cell wall and the cytoplasmic membrane. In step 2, the aminoglycosides bind to the ribosomes, resulting in inhibition of protein synthesis. So now, let us study in detail the step 1 in which the aminoglycoside is transported inside the cell. Now, the step 1 of transport of the aminoglycoside is a multi-step process. First, the aminoglycoside, it diffuses across the outer coat of the gram-negative bacteria. Suppose this is a gram-negative bacteria. So, in the first step, the aminoglycoside, it passes through the porine channels present on the cell wall of the gram-negative bacteria. Now, after entering into the cell wall, this aminoglycoside enters from the periplasmic space. This is the periplasmic space. Now, from this periplasmic cell, the aminoglycoside, it enters across the cytoplasmic membrane, and this transport is mediated by the electron transport chain. So first the aminoglycoside, it enters the bacterial cell wall through porin channel, and from this, and then enters the periplasmic space. From this periplasmic space, it enters into the cytoplasmic membranes. This green color is the cytoplasmic membrane. This transport across the cytoplasmic membrane is mediated through the electron transport chain. Now, this penetration is dependent upon the maintenance of polarized membrane, and this maintenance of polarization in the membrane is maintained by oxygen. Also, penetration is activated by increase in the pH. At alkaline pH, penetration is 20 times more than acidic pH. Now, as the penetration is totally oxygen dependent, the inactivation of the antibiotic occurs in anaerobic condition. So, in anaerobic condition, aminoglycoside do not work on the bacteria. Now, the step 2 occurs inside the cell, in which the aminoglycoside molecule, it binds to the ribosomes. Now, inside the bacterial cell, the aminoglycoside, it binds to the 30S and the 50S subunits at the 30S-50S interface. Now, this binding results in freezing of protein synthesis, prevents polysome formation, and it causes increased disaggregation of the monosomes. Increased distortion of mRNA codon recognition occurs, which results in misreading of the code, and wrong amino acids enter into the peptide chain. So, entire protein synthesis process is broken down by this aminoglycoside. Now, let us study the cidal action of the drug. The cidal action of the drug is based upon the secondary changes in the integrity of the bacterial cell membrane. Now, after exposure to aminoglycoside, the sensitive bacteria becomes more permeable to ions, proteins, amino acids, and through these permeable membrane, the ions, proteins and amino acids, they leak out, and because of this the cell death occurs. So permeability of the bacterial cell is altered by aminoglycoside, which causes leaking of the important macromolecules inside the cell, leading to cell death. Also, aminoglycoside induced alteration of the cell membrane augments the carrier-mediated entry of the aminoglycoside in the bacterial cell, resulting into lethal action. Now, let us study the mechanism of resistance of the bacteria against the aminoglycosides. There are various mechanisms of resistance. The first one is the acquisition of inactivating enzymes against the aminoglycosides. Now, if the bacteria acquires inactivating enzymes against aminoglycosides, what these inactivating enzymes do? They either phosphorylate or adenylate or acetylate the aminoglycosides, resulting in the formation of conjugated aminoglycoside. Now, this conjugated aminoglycoside, it do not bind to the target ribosomes. So, the protein synthesis is not hampered and the desired effect of the drug is not attained. So, this is one of the mechanism of resistance, that is acquisition of inactivating enzymes. Now, these inactivating enzymes are acquired by the bacteria either by conjugation and transfer of plasmids. Now, nosocomial microbes which are involved in the hospital-acquired infection are rich in such kind of resistant plasmids. So, the hospital-acquired infections are generally multi-drug resistant. Now, the second mechanism of resistance is by mutation. Due to mutation, there is reduced affinity of the ribosomal protein that normally bind to the aminoglycoside, resulting into high resistance. Third mechanism is reduced efficiency of aminoglycoside transport mechanism. The pores present on the bacterial cell wall becomes less permeable, so the active transport of aminoglycoside is interfered. Now, the shared toxicities of aminoglycosides are ototoxicity, nephrotoxicity, and neuromuscular blockade. Now, the precautions and interactions of aminoglycosides. The precautions to be taken is it should be avoided in pregnancy because it may lead to fetal ototoxicity. Also, avoid the concurrent use of nephrotoxic drug with aminoglycosides such as NSAIDs, vancomycin, amphotericin B, and cyclosporin. Cautiously used in later ages or in elderly patients because of risk of kidney damage. And aminoglycoside should not be mixed with any other drug in the same syringe or infusion bottle. Now, the pharmacokinetics of the drug. Aminoglycosides are highly ionized. They are unaffected in the GIT. There is rapid absorption from the intramuscular site. Peak plasma level is reached in 30 to 60 minutes. It enters the blood-brain barrier and placenta. It is excreted unchanged in the urine, and the plasma T half of the drug is 2 to 4 hours. Now let us study each of the aminoglycoside antibiotic in detail. The first one is the gentamicin. Introduced in the year 1964, it has high potency and broad spectrum of activity. It is the most commonly used aminoglycoside for acute infections, and it is the prototype drug of this group. The spectrum of activity is highly sensitive against aerobic gram-negative bacilli such as E. coli, Klebsiella, Enterobacter, Haemophilus, Proteus, and Serratia. Then, few gram-positive bacteria are also susceptible such as Staphylococcus aureus, Streptococcus facilis, Listeria, Streptococcus pyogenes, Streptococcus pneumoniae, and Enterococci. While the gentamicin is totally ineffective against tuberculosis. Gentamicin is more potent than streptomycin, kanamycin, and amikacin. It is equally potent to tobramycin, cecomycin, and netilmycin, and it synergizes with the beta-lactam antibiotics. Used in dentistry, it is predominantly against the gram-negative bacteria. It cannot act on anaerobic conditions, and dental use is very limited. The only dental use of aminoglycoside here to remember is gentamicin 2 mg per kg, either IM or IV, plus amoxicillin or vancomycin is given as prophylaxis against subacute bacterial endocarditis following dental surgery in patients with prosthetic heart valves. The general medical use of aminoglycosides include in respiratory tract infection in critically ill patient, burns, UTI, abscess, septicemia, meningitis, and the subacute bacterial endocarditis. Now the streptomycin, it is the oldest aminoglycoside obtained from Streptococcus griseus. It is restricted to the treatment of tuberculosis. It is less potent and has a narrow spectrum of activity. It covers aerobic gram-negative bacilli such as Haemophilus, Brucella, Yersinia, F. tularensis, Nocardia, and Mycobacterium tuberculosis. Adverse effects of this drug include auditory disturbances, nephrotoxicity, hypersensitive reactions, and superinfection. Uses in dentistry are not so common, but in general medical practice it is used against tuberculosis, plague, and tularemia. Now the next drug is the kanamycin. which was introduced in 1957, obtained from Streptococcus myceticus. It is the second systemically used aminoglycoside, but it has high toxicity. It is used in urinary tract infection and a resistant form of tuberculosis. The tobramycin, introduced in 1970, obtained from Streptococcus tenebrarius. Action is similar to gentamicin. It is less toxic and it is 2 to 4 times more potent against Pseudomonas. The amikacin, it is a semi-synthetic derivative of kanamycin. It is resistant to the bacterial aminoglycoside inactivating enzymes. It has a wide spectrum of activity and high doses are used in Pseudomonas, Proteus and Staphylococcal infection. Uses is in hospital-acquired gram-negative infections, bacillary infections and MDR-TB. Cesomycin, introduced in 1980s, it is a natural derivative of Micromonospora inyoensis. It is similar to gentamicin. It is potent on Pseudomonas and beta-hemolytic streptococci. Netilmycin, it is a semi-synthetic derivative of cesomycin with a broader spectrum of activity. It is resistant to the inactivating enzymes. The spectrum of antibiotic is against few gentamicin resistant strains, Klebsiella, Enterobacter, Staphylococcus, but it does not cover Pseudomonas aeruginosa. Neomycin, obtained from Streptococcus fradii, it has a wide spectrum of activity against all gram-positive and gram-negative cocci. Pseudomonas and asparagines appear to be resistant to it. Then, it is highly toxic to ear and kidney and it is poorly absorbed from GIT. It shows no systemic toxicity. Uses is in infected wounds, ulcers and burns, which can be used topically, and orally it can be used for preparation of bowel before surgery. And hepatic coma, neomycin, it reduces the intestinal flora and it reduces the NH3 production in the colon. Adverse effects of neomycin are rashes and damage to the intestinal villi. Framycetin, obtained from S. lavandulae, has high systemic toxicity and it is used topically for skin, eye and ear infections. So here we complete with the lecture on aminoglycoside. There might be some auditory disturbances in the background. So please cooperate with us. Please bear with us for this time. Next lecture will be on macrolide antibiotics. Please subscribe to our channel and happy studying.